Okay, let's finish this set of notes. I already did. I already started recording. Where we were last time was we were talking about the fact that, and it's been ages, so we can like review it, right? It's been like a week. You guys are going to be a little ahead of the last class because we had a medical incident in the last class that took like half an hour, right? I think it took like half an hour. Yeah, so I'm going to have to try to get you guys back on track with them. So where we were before the break was that we were talking about if the body-wide goal is homeostasis, but cells are specialized, then it necessitates communication, right? Remember? And so we said that the mechanism by which you communicate between cell A and cell B is chemicals, right? So if I'm cell A and you're cell B, I can release a chemical that you can respond to as long as you have a what for it? Exactly. So that would make her a target cell for that chemical messenger. And the thing that communicates between cell A and cell B is called an intercellular, yes, between cells, chemical messenger, because that's what it's actually doing. So today what we're going to do is figure out how to categorize and classify the chemical messengers. Before we do that, I just want to make sure that you understand the concept of target cell and receptor, which you should have, of course, learned in 1610, right? Anything with a receptor for what I'm talking about will be considered a target cell for that. But does that mean you're the only target cell for that chemical messenger? Or could she also be a target cell if she had the same receptor, right? It's not limited to a single target cell, right? But then the other thing, and I just want you to have heard this term, even though we're not going to go into it in a lot of detail today, is that just because the chemical messenger binds to the receptor on the target cell hasn't actually responded yet, right? So there is a mechanism by which the target cell has the response. So let me give you an example. If a chemical messenger is released and binds to a receptor on you, and the chemical messenger's job is to open a channel, then you just open the channel. It's like a one-step process. But what if the chemical messenger, what if I am a testis cell, and you are a skeletal muscle cell, right? And I'm going to release a chemical messenger called testosterone. And testosterone causes skeletal muscles to do protein synthesis of actin and myosin, the structural contraction proteins in skeletal muscle. But you guys know enough chemistry and enough biology to know that protein synthesis is not an instantaneous one-step process, right? It's a whole series. You have to go to the nucleus, have to initiate transcription and then translation and all of that. There's a whole bunch of enzymes and proteins involved in the whole process. So the process by which you get this target cell to respond is called the signal transduction pathway. And it's not magic. Cell biology, right? And it can be really long or really short depending on what you're trying to get the cell to do. What we will do later on in the semester is we'll start fleshing out the signal transduction pathways. But right now I'm saying just because I tell you to do it, it doesn't mean poof, you do it like magic. It's still cell biology. Okay, but we'll come back to that later in the semester. So what I'm going to talk about now is these chemical messengers, these intercellular chemical messengers that you use to communicate between cells. How do you categorize them? There's a bunch of possible ways to categorize them. You could categorize them chemically. You could say, I'm going to put all the lipid ones in one category and all the protein ones in one category and all of the amino acid ones in one category. That's not how they do that because it doesn't actually pertain directly to functionality. This is all about physiology, which is function. So instead, how you're going to categorize them is how they get from point A to point B, okay? So if I get dumped into the bloodstream and travel through the bloodstream from the cell that releases me to the target cell, then I'm going to be much slower than something that is released right between the target cells and just diffuses, right? Does that make sense? So functionally, they're going to fit better into categories based on how they travel from point A to point B. Does that make sense? So the first category are going to be the ones that travel through the bloodstream from point A to point B. And that means that they're probably not going to have an instantaneous response. Does that make sense to you? If I am released by the testis, and I'm trying to cause a response in this skeletal muscle up here, then you are not going to see it occur within a matter of nanoseconds, right? But if I am released by a cell to communicate with the cell right next to it, you can have an almost instantaneous response. So the first ones, the ones we are going to talk about first, are the ones that are released from a cell, get picked up by the bloodstream, travel through the bloodstream from point A to point B. And you already know this word. These are categorized as hormones. And so hormones, here's a visual on it. Here's the cell that releases it. Gets dumped, gets almost immediately picked up by the bloodstream and travels through the bloodstream to get to the target cell with the receptor. Now, these guys, of course, are probably not going to have an instantaneous response to the release, right? In addition, are they limited in how many target cells they can actually interact with? If you dump something into the bloodstream, how many places could it possibly go?
Millions.
All over the place. You could have receptors on almost every single cell of the body. So I just want you to think again about testosterone and think about either being a boy going through puberty or knowing a boy going through puberty. And if you want to see what testosterone does and how many places it has a receptor, just look at the changes that occur in a boy going through puberty. Because if testosterone, the biggest change during puberty is ultimately going to be the spiking of testosterone in a male's bloodstream. There are other changes, but that's the biggest one that causes massive changes. So testosterone goes up, but it has an effect on muscle mass. Yes, we've already talked about that. What about bone?
Yeah.
Bone, yes. Does it have an effect on hair follicles? Yeah. Oil? Sweat? Yeah. So all of those, you're telling yourself right now that those are all target cells for testosterone. They all have receptors for testosterone. And let's not ignore the completely obvious that testosterone has receptors on your brain. Does it not? Right? My husband tells me that he has never been the same since puberty. He's like, I used to think about Legos, and now I just try to take everything and make a boob out of it, right? And he's like, I wish I could just turn it off sometimes. I think I'd gain 20 IQ points if I could just turn this crap off sometimes. But the same thing with women. With women, you get estrogen, and you can see all of the changes that occur in the body because of estrogen. So what you're saying is that if I dump it into the bloodstream, there's no limit to the number of receptors. Insulin, it's not a reproductive hormone, but insulin has receptors on almost every single cell in the body because almost every single cell in the body needs to take in glucose in order to do cellular respiration, right? So those are all target cells. So it's going to have potentially very broad impacts, but not very quick impacts. In addition, you might not see it turn on immediately, but you also don't see it turn off immediately. So whenever, for instance, something is released and dumped into the bloodstream, you can't say, oh, I'm done with it, because then you have to wait for it to get out of the bloodstream, right? To get broken down via the liver or the kidneys or the target cells or whatever breaks it down. So usually, as far as the impact that you would see, it'd be kind of a slow upsweep, right? And then a slowdown, right? And potentially last for long periods of time. So hormones can have widespread effect. Hormones allow for what we call endocrine communication. Now I want to back up and put this back together with the stuff that we were doing in the last lecture. And if you are dumping something into the bloodstream and it's released by the endocrine system and I'm using it to try to accomplish negative feedback, would that fit into the category of extrinsic or intrinsic regulation? Extrinsic. Do you remember me telling you that extrinsic generally uses the endocrine system or the nervous system, right? So reproductive system using the endocrine system to try to maintain homeostasis is extrinsic regulation. Does that make sense? Okay, so some examples of hormones. Some of them are reproductive. If you aren't taking physiology or aren't going this far into it, generally all people think of about hormones is reproductive ones. Testosterone, estrogen, progesterone. But you know, of course, there's way more hormones than that. You've got cortisol and insulin and glucagon and thyroid hormone. Can you guys think of any more? Any other hormones? Yeah, GnRH. Yeah, good. ADH. Good. Any other ones? Thyroid, parathyroid. By the time the semester's over, you'll know at least 32 different hormones, where they're coming from, where they go to, what they do, what happens if they're broken, all that kind of stuff. And that's a subset of the hormones that we have, just like enough to get you started on the basic understanding. So your textbook uses another term, and I need you to know it, and then I'm really not going to use it for the rest of the semester. Your textbook says that there's a special category of hormones called neurohormones. And neurohormones, they say, are released by a neuron. So if you just made this cell right here, a neuron, and then dumped it from a neuron into the bloodstream to travel to its target cell, then they would categorize it as a neurohormone. But from my perspective, who the hell cares what kind of cell releases it if it gets dumped into the bloodstream and travels to a distant cell? So with endocrine, a lot of times we're used to thinking of endocrine organs as glands, like the thyroid gland and the pancreas, but they're not all glands. For instance, the heart releases a hormone. You can have fat that releases hormones. So there are a bunch of different kinds of cells that are releasing hormones into the bloodstream. So your textbook will call the ones that are released by neurons and get dumped into the bloodstream neurohormones, and they include ADH and oxytocin. But in my mind, they go in with hormones, and that's how I categorize them, because they're going to have a slow upsweep, a slow downsweep, and potential for widespread effects on the body. The reason that I like to lump them in with hormones is because I don't want you to confuse them with the next category, which is neurotransmitters, which is going to behave differently. Okay, does that make sense? Okay, so let's talk about neurotransmitters. And neurotransmitters are next. Neurotransmitters have to meet two criteria, not just one. They have to, yes, be released by a neuron, okay? But they also have to be released right next to the receptor, okay? Because if they were released by a neuron and dumped into the bloodstream, then they would be categorized as what? Hormones, exactly. So they have to be released by a neuron right next to the receptor. This allows for what we call neural communication or synaptic communication. The site at which a neuron communicates with the next cell, you should have learned in anatomy, is called a synapse, right? And neurons are very limited in the types of cells that they can communicate with. They can communicate with other neurons, but they can also communicate with all three types of muscle, right? All three types of muscle can be effectors for neurons, and also they can communicate with glands. But notice I didn't tell you that about hormones. Are hormones limited to the types of cells they can communicate with? No, hormones can communicate with pretty much anything, right? Okay, so a neurotransmitter compared to a hormone. Based on this release mechanism, what do you think about the speed of target cell response compared to a hormone?
Very quick.
Very quick, exactly. But broad? Is it really broad? Is it going to impact lots of different cells simultaneously? No, it's very quick and it's very tight, right? Very tightly controlled, very pointed, right? Does that make sense? Okay, so it's going to be a really quick on. It can also be a really quick off, because I don't have to wait to get this out of the bloodstream. I can just remove a couple of pieces out of the synapse and then I can turn it off. So quick on, quick off, not broad, okay? But this one is also which category of regulation. If I'm using neurotransmitter release to maintain homeostasis via negative feedback, is it intrinsic or extrinsic regulation? It's extrinsic too. It's faster extrinsic, but it's still extrinsic, okay? So let's talk about some neurotransmitters. Communication is faster but shorter lived than endocrine. They don't go into the bloodstream by definition. Examples of neurotransmitters, you've heard of them because either you've been depressed or anxious, or you know somebody who has, or you've had some kind of mental challenge, or you know somebody who has. So your happy chemicals, if they're all perfectly balanced, and I might have had like 10 minutes in which mine were balanced ever in my life, norepinephrine, dopamine, and serotonin. And you got a whole bunch that are not necessarily related to that. You've got acetylcholine, you've got a bunch of other ones. We are going to learn a whole bunch of neurotransmitters this semester. This is my particular passion. I love neurotransmitters and brain function. So you're going to learn an awful lot about them this semester, and you're going to learn the best, most current information that I can possibly give you about neurotransmitters and drugs and those kinds of things. So we will spend a decent amount of time talking about those. And then the ones that are probably the least familiar to you but are the most common in the body are the paracrine agents. And paracrine agents are released by a cell to communicate with its neighbors. So I want you to imagine that everybody on this front row is they are all part of the same tissue. And so they're going to be the same kind of cell or similar kinds of cells, right? And so I want you to imagine that she has some kind of injury or infection. And she needs to tell her neighbors, right? She needs to give them heads up that there's something around, there's something happening. But does she need to tell the entire body? She doesn't, right? She needs to just tell her homies at first. We're going to try to handle this in-house. So what's going to happen is she's going to release a chemical that can communicate with her neighbors, right? This chemical is going to be for local communication, okay? And what does that sound like to you of the examples that we talked about before, extrinsic or intrinsic regulation? This is intrinsic, and remember me telling you that almost every single cell in the body had the capacity to try intrinsic regulation? Almost every single cell in the body has the capacity to release paracrine agents. So what she's going to do is, let's say she's got some kind of pathogenic infection, got some kind of viral infection, and then she's going to release something that says, hey, heads up, there's a virus around. And her friends are going to go, cool, thanks, dude. They are going to try to manage it, right? And they're going to try to manage it by a bunch of local things, and the bunch of local things may be, yeah, let's do some local vasodilation, increase blood flow to the area. Let's increase macrophage activity in the area. Let's cause any kind of immune system response. Let's constrict something. Let's dilate something. Let's increase smooth muscle activity or decrease smooth muscle activity. Any number of local things. What if you're injured? You could release something that says, let's clot, right? Don't those all sound like things that you want to try locally before you ever try systemically? Yeah? So these are all paracrine agents. So let's look at some examples of paracrine agents. So this is cells within a tissue communicating with other cells in the same tissue generally. And with this, you need to generally release very low concentrations into the interstitial fluid, right? Does that make sense to you that you wouldn't need to release anywhere near as much if you were just going to communicate locally? Which one of the three, paracrine, neurotransmitter, or hormone, do you think that you have to release in the highest concentration in order to get a response? Yeah, hormones. Because when you dump it in the bloodstream, you have no control over where the hell it goes. If she's got a receptor for it, does that mean I'm always going to bind to it, or I might just go, hey, on my way by, right? In addition, I'm like going to have to go through the liver, and some of it gets broken down, gets peed out of the kidneys. So a lot of times you have to like blanket bomb when you're like trying to do hormones. But when you're releasing paracrines, you just have to release small quantities, at least at first, right? If it works at first, then you would just release small quantities and stop. So small quantities released into the interstitial fluid. The mechanism of getting to point A from point A to point B is just generally diffusion. You just diffuse between the 2 points because it's, you know, microscopically separated from one another. And then almost every tissue, almost every cell in the body has the capacity to release paracrine agents. Now, is it possible at all that you can imagine that, let's say, okay, you get the same infection, I've got a viral infection, and I release something, I'm trying to tell my friends that I've got a viral infection, but it doesn't take care of the viral infection. Then my friends get viral infections, and then they're releasing paracrine agents, right? And so we started out on the front row with our viral infection, and then it spread all along the front row, and I'm still failing, because sometimes you just do, right? And then you guys have it, and you're releasing paracrine agents, and you guys have it, and you're releasing paracrine agents. If you release enough of anything, it will get picked up by the bloodstream. So is it at all possible that something that started out respiratory can cause systemic responses like fever or sedation? Yes? If you release enough of it, you're going to get a systemic response, okay? So, or can I have a nasty wound that is not healing that can actually cause me to have a fever? Yeah. So it's still paracrine agents. It's just this sort of second-tier response to paracrine agents. They don't always go there. They don't even usually go there, but they have the capacity to go there. If you release enough of them, then you can make yourself systemically impacted by the release of local chemicals in high quantities. Does that make sense to you? So there are tons of different kinds of paracrine agents. Let's see if some of these make sense to you. There are more than you could possibly memorize. I want you to know a few, some of them because they're going to be relevant in the rest of the semester, and some of them because you're already messing with them, so you might as well know what the hell they are. So does it make sense to you that clotting factors would be a local or paracrine activity? You certainly don't want to clot your entire bloodstream, right? That would be your worst day ever. It would be your last day ever, actually. Growth factors. What if I damage something and I need to go through cell division? I don't need to do it body-wide because I don't need to grow a foot, right? I need to do it locally. So growth factors tend to be paracrine agents. Histamines, if I have an allergic response, I take in something, the allergic responses will tend to cause vasodilation. So let's say we start out with an allergic response. I am allergic to poison ivy, and I get exposed to poison ivy. I will release local paracrine agents, right, which will cause vasodilation, increase white blood cell activity in the area, all of that. That is local. Now, could it happen that I was so massively allergic to poison ivy that I could also go into anaphylactic shock because of That's the second tier response. Do you see that? If it locally is just supposed to cause a little vasodilation, but then I release massive amounts of it, then I do massive vasodilation and my blood pressure drops. Does that make conceptual sense to you? It's certainly not plan A, and it's not how it always happens, but you can put it together and tell that story, right? That's why the kid at school cannot be exposed to peanuts, right? Because he doesn't release just a little bit of those vasodilators, massive amounts of those vasodilators that send the kid in shock, okay? Okay, so, and then cytokines. Cytokines are the ones that are the most likely to get dumped into the bloodstream and cause a massive systemic response. They don't always do that, but the second tier response is not uncommon. They are released from white blood cells, among other cells in the body. Does it make sense to you that white blood cells are super duper good at releasing paracrine agents? Because isn't that what they're generally first trying to do? Like start something just in this area, not everywhere? So these are very likely to get dumped into the bloodstream if you don't respond very quickly to what's going on. And the only reason I mentioned these is because there's some new therapies that you may or may not have heard of that actually use these paracrine agents. So we figured out how to make these paracrine agents in the lab. And among other things, what they do is they're part of the body's immune response against pathogens and also against cancers. And so what we did is we figured out how to make some of these cytokines in the lab. Specifically, two categories of them called interleukins and interferons. Have you guys ever heard of interferon therapy? Okay, so interferon therapy is used in a couple of different ways. Initially, the first time that I heard of it, it was used for therapy for HIV to keep it from going to full-blown AIDS. So if this is an immune system release, right, that is helping you to battle off the pathogens in your body, and I can artificially give you some interferons, right? Can you see that that would reduce the likelihood that you would go from HIV to full-blown AIDS if I can keep the viral load really low? It's one of the many, many drugs that you can use with HIV to keep it from going to full-blown AIDS. It's very expensive. It's not readily available. One of the many examples of the fact that you get better medical care when you're rich than you do when you're poor. And the other place that they're using interferon therapy, which is more recent, is that they've developed some interferon therapies for certain forms of cancer. Yeah. It's not all forms of cancer because different cells respond to different interferons, right? But there are certain forms of cancer that we actually know which interferon will interfere in their cell division process, right? And so you can use interferon therapy to keep cancer from metastasizing, to keep it from growing. Does that make sense? And it's all based on these paracrine agents. Good? Okay, so those are interferons. And then the last category are the ones that you mess with all the time, even though you don't know that you're messing with them all the time. And these are called eicosanoids. Will you say that, please? Eicosanoids. It's a huge category. A bunch of different cells release them. They do all kinds of different things. In the stomach, they can increase or decrease gastric secretion. They can dilate or constrict your bronchioles. They can cause vasodilation or vasoconstriction. They can shrink your nasal membranes to make your nose run. All of those are like paracrine activities. So let's look at some of them. So this is a major category of paracrine agents, and they're all derived from arachidonic acid, which you may or may not know, but I know that you know phospholipids. Yes? How many cells have phospholipids in their membranes? How many of your cells? All of them. All of your cells have phospholipids in their membranes. So if I tell you that these are all derived from phospholipids, so here's your little phospholipid. Looks like a little M&M guy, lost his arms, right? Okay. This is arachidonic acid, which is their intermediate molecule. And then all four of these right here are the eicosanoids. Okay, so why do you care about those? The reason you care about those is because you mess with them. You mess with them when you are trying for pain relief, anti-inflammation, and fever reducing. So let's talk about each one of them. This one right here is leukotrienes. Leukotrienes, just from the prefix, I bet you can tell me what kind of cell is likely to release it. Leuko? White blood cell, yeah. White blood cells are the ones that primarily release leukotrienes, and their job is primarily inflammation and also the initiation of immune response in an injured tissue. Inflammation is not bad. Inflammation is just fine when it's acute, right? It's the first step of healing when it's acute. Everybody with me? When it's chronic, it can cause problems of its own. Or if it's in an inappropriate place like a closed head wound, of course it can cause problems of its own, right? But inflammation is sometimes painful. So we still treat it even though it's not necessarily always negative, right? Because you don't really want to be in pain. So keep that in mind. So that's your leukotrienes, and those are this part of the story, okay? And then the other ones, prostacyclins and thromboxanes, those two at the bottom, those are involved in blood clotting, which should also be a paracrine activity, correct? Okay. So blood clotting. And then prostaglandins are the biggest category, and prostaglandins are the ones that do a ton of different things. All those things that I was listing before are among the functions of prostaglandins. There's at least 16 different kinds. They're released just freaking everywhere. And they can do any number of things. Like I said, raise lower blood pressure, gastric secretion, nasal membranes. But please look at what they do when you release them in that second tier quantity. They cause sedation and potentially fever when you release enough of them. So again, that's not the first thing they do, but if you keep dumping them, right? Keep dumping them. They get picked up. They can cause a fever, or they can cause you to be exhausted, which is sedation. Okay. So now let's go to why we're talking about those things. So now we've got inflammatory, potentially pain, right? Okay. We've got blood clotting, and we've got more pain, potentially sedation and fever. Do you see that? So because you take chemicals when you have these kinds of things, you have messed with these before. So let's talk about pain relievers, anti-inflammatory drugs, and eicosanoids. So aspirin and the category of drugs called NSAIDs, non-steroidal anti-inflammatory drugs, which includes ibuprofen and naproxen sodium, and kind of, but not exactly, acetaminophen. Acetaminophen fits into the story, but not quite as neatly. It still kind of does. But so think ibuprofen and aspirin for right now. What those do is those block the synthesis of many of the eicosanoids, including the prostaglandins. And because they do that, they reduce pain, fever, inflammation, right? Because they block right here. They block cyclooxygenase. Okay, that makes sense? But in addition, they also, of course, inhibit clotting. You guys know that if you have any kind of heart problem or cardiovascular problem, sometimes they use a maintenance dose of aspirin to reduce your likelihood of inappropriate clotting. So I have atrial fibrillation and a murmur. I'm on a daily 80 milligram aspirin regimen, which keeps me from inappropriate clotting, right? It also keeps me from appropriate clotting. So if anybody punches me, I end up getting the hugest bruise ever. So I just don't hang out with people who punch me, generally. It does that. And do you also know that if you're taking any kind of blood clot preventer or blood thinner, that you're not supposed to also be taking over-the-counter aspirin or ibuprofen? You know that, right? Okay, so that's that half. But that still leaves us this inflammatory response, right? Okay, so, oh, by the way, there are prescription forms of cyclooxygenase inhibitors. You can't get those over the counter, but you can get them prescription. You probably heard of them before, Vioxx and Celebrex. They're COX-2 inhibitors. They're really powerful pain relievers and used sometimes for rheumatoid arthritis or chronic inflammatory conditions. And they're more powerful, but generally when you're more powerful, you're going to have more side effects. And there was actually a big lawsuit associated, I think, with Vioxx, wasn't it? Is it Vioxx? Yeah, there's a big lawsuit. It's not dangerous for everyone, but there's certain categories of people that shouldn't be taking it. Okay, and then what if I want an even more powerful anti-inflammatory? If I want a more powerful anti-inflammatory, I could inhibit here, right? Phospholipase A2. And then I would inhibit all of those inflammatory responses plus all of these, right? Okay, so we have the most powerful anti-inflammatories that we have ever found are sometimes referred to as steroids or corticosteroids or adrenal steroids. And you may have heard of them. You've probably heard of prednisone, right? Cortisone, corticosterone. Those are really, really powerful anti-inflammatory drugs. And they are not used as maintenance medications. And for reasons I'll explain in just a second. But if, for instance, I had a closed head wound, then it's the inflammation that is life-threatening, correct? Right? Then you can use a really powerful anti-inflammatory. Or what if I had a child that had such serious chronic life-threatening asthma that instead of just using a bronchodilator inhaler, they also used an anti-inflammatory inhaler, a prednisone or a steroid inhaler. Does that make sense? Now, I don't know if you've ever had a kid or known a kid. They don't keep people on those for very long. They put them on and then they take them off. And the parents are like, it's working, why the hell are you taking it off? I'm going to tell you why they're taking it off. Because what they use, and this is a little hard to wrap your head around, but it's interesting and pertinent. The drug they use to inhibit phospholipase A2, okay? That's the drug, not the phospholipase, okay? So these are all paracrine agents, you guys with me? But the drug that they use to inhibit phospholipase A2 is a mimic to a hormone in your body. The hormone called cortisol, which is a metabolic hormone, okay? So can you see what widespread potential side effects you would have when you're taking large quantities of a hormone, right? So why the hell would you do it? Because death is the other option. They don't use these drugs lightly. They only use them in a situation in which you have such life or quality of life threatening inflammation that you have to. And then they put them on and they take them off, and they put them on and they take them off. Does that make sense? Okay, so I'm not saying that any of these are hormones. I'm saying that the drug that you use to inhibit this is actually a hormone. It has massive, have you seen the side effects from using, for instance, prednisone for long periods of time? It's a metabolic hormone. It'll cause you to have massive amounts of weight gain, massive amounts of water retention, really affects your behavior, makes you angry, break out everywhere, completely trashes your metabolism. It's not a good one to go on if you can avoid it. Okay, so those are the ones that this whole inflammatory response that we're talking about all has to do primarily with paracrine agents, right? The whole inflammatory and immune system response. Okay, so the last thing in this set of notes, the last thing on that is, what about an autocrine agent? So we said we've got hormones released into the bloodstream, neurotransmitters released from a neuron right next to the receptor, paracrine agents released within the same tissue. What about an autocrine agent? Well, an autocrine agent is simply acknowledging that if I release something that communicates with my neighbors that are the same cell, same type of cell as me, I probably have a receptor for it too, right? So can you see that if all of these guys are the same kind of cell and she releases something that can communicate with her, that it's not impossible that I could go back and tell myself something, right? When you do that, it's considered autocrine. Does that make sense? But, huh? You just can't not tell yourself something. If you release something and you have a receptor for it, sometimes it binds to your own receptor. It's not like you're trying to do that particularly, right? But if these two are releasing things that can communicate with each other and they both have similar receptors because they're similar cells, then sometimes you can end up telling yourself the same message. So paracrine and autocrine in my mind go together because generally it's the same chemical that could communicate with you, or I could actually use it to communicate with myself, right? And it's generally not released to communicate with yourself, but it does because I have receptors for it, right? So it's not like you did extra to do it. You're just like dumping it and I might have a receptor and it binds to it because it fits. That's it. Okay, so that's autocrine. So in my head, I usually dump paracrine and autocrine together, okay? Okay, so is there any situation in which I could communicate between cell A and cell B without releasing anything into the extracellular fluid? And the answer is yes, if the cells are stuck together, right? Like gap junctions. Remember learning about gap junctions in anatomy? So when you do that, if cell A and cell B are stuck together via gap junctions, right, which is just two channels that are actually stuck to each other, then cell A and cell B are going to have instantaneous communication with one another, but not varied communication, because cell B will always have to do exactly what cell A did, right? So you can't have them behaving differently if they're both stuck together and responding to the same chemicals. So although it's instantaneous, it's very effective, right? It's like a one-trick pony. You can only like do the same thing that your neighbor did. So not surprisingly, it's not super useful in the human body. I mean, we do have gap junctions, of course, in cardiac muscle, and we have them in our respiratory epithelium, and we have them in smooth muscle, but we don't have them in that many places. There's not that many situations in which you want every single cell in a tissue to behave exactly the same way. Sometimes you want them to have specialized functions. So for some reason in your textbook or in physiology textbooks, they decided to change the name. They're like, oh, we're not going to call them gap junctions anymore. We're going to call them connexons. Somebody just made that term up. They're like, you know what, I want to name it a connexon. It's a cool word, right? I don't know. It's always been that way in phys books as far as I know. And anatomy books, they always call them gap junctions. Those are gap junctions, just so you know. Same damn thing you already learned. They're just trying to confuse you. Okay, so you don't see as many of those as you would imagine. In an adult, they exist those places that I talked about. You do have them, interestingly, in the synapses in an embryo, embryonic nervous system. I don't know why I find this interesting. When you're putting together your nervous system, when it's first put together, and you're sort of like putting all the parts together as sort of a model of the connections of the nervous system, all the cells are stuck together via gap junctions. And so they're all just sort of functioning in the same way. But then when you start needing it to act like a brain, right, and have some functions different than others, they get pulled apart and then they behave as synapses instead of just like gap junctions. So you don't have very many in an adult. Okay, so the last concept before we leave this. How do you determine whether a chemical messenger that you are looking at is a neurotransmitter, a hormone, a paracrine, or autocrine agent? Some people like to memorize them. They like to say, hey, dopamine is a neurotransmitter. That's not a good way to do it in physiology. What you need to do is look at what it's doing. If dopamine is released from a neuron to communicate with another neuron, then it is a what? Neurotransmitter. But if dopamine is released from a neuron and dumped into the bloodstream, then it is a what? Hormone, exactly. So when somebody asks you, is dopamine a neurotransmitter or a hormone, your answer is yes. Because the body can do any damn thing it wants with a chemical messenger. And interestingly, once the body learns how to make something, it can use it in multiple ways. It can say, I'm going to release this as a neurotransmitter here, but over here I'm going to dump it into the bloodstream. Okay? So the point there is that memorization will only get you so far in physiology. It gets you to the point that, like, okay, I know that dopamine is released in these parts of the brain. It's generally a neurotransmitter, but don't use the rules at the expense of the truth, right? The truth is that you look at what it's doing, and then you decide what it is, and then quite often you will see it behaving different ways in different circumstances. Does that make sense to you guys? Okay, so that is it for that set of notes, and then we will start on the next one. And the next one has a lot of overlap between 1610, but I'm just going to focus on different parts of it, really. Okay. So this is chapter 2 and 3, and it has a lot about cell structure and function. But here's not what we're going to do. We're not going to go through and say this is the ribosome and it does protein synthesis. This is the Golgi apparatus and it does processing and packaging. And this is the nucleus, because you already know that. And if you're not confident with it, then you need to go back and do this before you begin section, in which you remind yourself what the basic anatomy and function of the cell is. And then you also need to remind yourself about some basic chemistry. And then after that, before you begin section, that's where I'm going to assume that you are at this set of notes. But what I need to do here is that primarily I'm going to use this set of notes to take the metabolism stuff that seems completely irrelevant to everything when you're learning it in 1610 and somebody's trying to teach you cellular respiration. And you're like, ugh, what the hell does this have to do with anything? Why are you making me learn this? We need to make it relevant to physiology because it's going to come into play the entire semester long. And just so you know, I can empathize because I tried as hard as I could to not learn cellular respiration over and over again, but this is where it came to bite me in the butt, was in physiology. I'm like, oh, it's totally relevant. Carbon dioxide, yeah, I get it, it was produced here. So we're going to talk about metabolism here. And we're going to talk about metabolism, meaning sort of the original meaning of metabolism, not I hate that girl because she's got a faster metabolism. But we will also put that back together, right, with what it has to do, cellular metabolism, body-wide metabolism. So metabolism really, from a physiological perspective, is all of the chemical reactions that occur in a cell, including the synthesis and the breakdown of organic molecules, the energy that is released and the energy that is used, right? All of that is going to collectively be involved in metabolism. And of course, cell-wide, you will have different levels of metabolism depending on the activity of the cell, and then body-wide, you're going to have different levels of metabolism. Before we go any further, we have to make sure that you know the terms anabolism and catabolism. Do you remember what those mean? You've learned them before.
Anabolism is breaking down, anabolism is building up.
Exactly. Catabolism is breaking down, larger molecules, usually, to smaller molecules. Metabolism generally, not always, but has the capacity to release energy. When you're taking something large and you're breaking it down, you have the capacity to release energy. Okay. Anabolism is taking small things like an amino acid and using that to build bigger things like a protein. And conceptually, even if you don't have it memorized, is that more likely to require energy or release energy? Yes, exactly, right? So if I'm taking something huge like a building and breaking it down to bricks, right? It is generally going to be an energy releasing reaction. If I'm taking something really small and making a freaking building out of it, it's going to generally require energy. And so, of course, what generally happens is that quite often in the body, you will couple a catabolic reaction with an anabolic reaction. The classic, most important, arguably, catabolic reaction is taking a nutrient like glucose and breaking it down to, for instance, carbon dioxide and water and releasing ATP as byproducts, right? That makes sense? So that is, of course, cellular respiration, and that's what we're going to talk about. But before we get there, we have to talk about enzymes and enzyme function, make sure you understand enzymes and how they play into this. So when we're talking about enzymes, what enzymes are going to do is, I hate the way they teach it in chemistry, and I think it's probably part of the reason it took me a long time to get it. When you learn about enzymes in chemistry, they always say that it lowers the activation energy, but that was always a little conceptually difficult for me because they would lower the activation energy for both catabolic and anabolic reactions. I'm like, well, I'm confused. You just told me that that one doesn't actually require any energy, and now you're telling me I'm using something to lower the energy, and I'm like trying to fit it, but they're using the terms entirely separately. But there is an easier way to think about what enzymes do. They make it more statistically likely that a chemical reaction is going to occur. Okay, so here is a way to think about enzymes, which has directly to do with physiology and binding sites and everything. So if this is the chemical reaction that I want to occur, I want A plus B to go to A bonded to B, okay? And A and B both exist inside a cell, for instance. I could just wait until they run into each other, right? And then I'm going to get very low levels of the product over time, right? If I just wait until they run into each other. Does that make sense to you? If I'm just like sort of waiting for them to smash into each other? So before we go into any further, make sure that you know the terminology that goes with these. You've learned them before. If you're doing a unidirectional chemical reaction, then these are the reactants, right? And these are the products, okay? Now, how can I increase the likelihood that A and B will form that product? I can provide a location that is attractive to A and attractive to B, right? A simple location. So if A is this shape, yes, and B is this shape, then what if I have a protein that has a binding site for each, right? I have just dramatically increased the statistical likelihood that I will get the product that I want. And the more enzyme that I have, and this is the enzyme, the more product I will get. Do you see? Okay. So this corresponds to what you've learned about enzymes before. Do you see that the enzyme is actually like not directly involved in the chemical reaction? Just sort of a landing space for the chemical reaction to occur. It's providing a location where the chemical reaction will occur. So it's increasing the rate of a chemical reaction over time, right? Making it more likely that it's going to occur. So you're going to get way more A and B if that occurs. So you've got a binding site for A and a binding site for B. When you are using reactants and an enzyme, then the reactants are now considered substrates. And this, right, is the what? What is that site called? This is called the active site where the substrates bind. You knew that. Good question, yeah. Do you know? I know. I do know. So let me show you that. You've seen this before, but let's just put it together. Okay, you haven't seen this figure before. But here's what people keep telling you over and over about the enzyme actually not getting used up during the chemical reaction, because it's really just a location where the chemical reaction occurs. So this one just happens to have one active site. Don't let that confuse you. But here's what's going to go. Here's the enzyme. It's got an active site that fits the substrate, correct? And with proteins, it's all about shape, right? Shapes tell you interactions. If the shape changes, then the function of the protein changes. So I've got an enzyme that fits the substrate. When the enzyme fits the substrate, a chemical reaction occurs that changes the shape of the substrate, yeah? And then it doesn't fit the substrate binding site anymore. It doesn't fit the active site. So it falls off, and now you got another enzyme that you can use over and over and over again, right? So this is the location where the things occur. So if you want a chemical reaction to occur, right, like forming a good marriage that your mother will approve of, right? The enzyme that you go to is church, yes? And the two things go together, and then you come out the other side, right? And you get the desired product. If, however, you would like to hook up, right, you go look for a different enzyme because you're looking for a different product, right? Same two things, different enzyme, different product. Does that make sense? Okay, so this is a shape-specific binding site. All right, so the substrate combines with the enzyme at the enzyme's active site. Importantly, the enzyme can be reused. Can it be reused infinitely? Nothing can. It can be reused a bunch of times. How many times is a bunch? I don't know. Probably different for you than it is for me. I know that I didn't get to make all of my enzymes once when I was a zygote and then I was done, right? I still have to replenish them. But I don't have to replenish them at every use, right? I have to make them infrequently. So enzyme activity, I want to talk about what can impact the rate of a chemical reaction, the rate at which you form your products. And there's some things that are intuitive and there are some things that are counterintuitive, and you kind of need to understand all of them. So the one, enzyme activity, and therefore the rate of chemical reactions can be affected by this concept. And you probably know this concept from either chemistry or 1610, saturation. Okay, saturation has to do with how many enzymes there are, right? And whether they're full or not. So let's say you have one enzyme and boatloads of A and B, right? Then the enzyme is going to limit the rate at which you get your product. Does that make sense to you? Because what's going to have to happen to each A and B? It's going to have to wait in line, right? It's like there's only one door to go through, right? And you just have to wait your turn to get to that door. So saturation, what is physically happening at saturation? Look at the graph. Here's the substrate concentration, and as the substrate concentration goes up, the reaction rate goes up, right? Until what? Until what? That's saturation, but I want somebody to tell me in your words what is happening physically at saturation. What? Reaction stops. Why? What is physical? Yeah, the enzymes are full. Exactly, right? And you couldn't change saturation just by adding more substrate, could you? No, they're still going to be full, right? What's the only way that you can change the saturation rate? Adding more enzyme. Adding more enzyme, is that an instantaneous thing? Is protein synthesis instantaneous? No, it's not, right? So the only way to change the saturation rate is to change the concentration of enzyme. So if you change the concentration of enzyme right down here, you can change the saturation rate, right? Okay, but that's the only way you can change the saturation rate, is to change the enzyme concentration. Okay, so keep that in mind. And now let's talk about a couple of other things. Substrate concentration is pretty obvious. Let's talk about that one. So what if I add more substrate? If I add more substrate, yes, then my reaction rate goes up until what? Saturation. Until I hit saturation. So substrate concentration does have an impact on the reaction rate, but only until you hit saturation. After saturation, we know the way to increase your reaction rate. Okay, so the next two sound very similar, but they're different. So you have to listen to the differences between them, and you may or may not have heard of these before. This one is called substrate specificity. Okay, I know that people tell you that there is only one thing that fits in a binding site, but they're only kind of telling the truth. Because there are a lot of chemicals that have a similar structure, right? And so maybe you have your favorite substrate right here, yes? And that one fits very quickly and very well, yes? And so you're going to get a very fast response when the substrate fits the enzyme really well. But in a pinch, in the absence of substrate C, maybe substrate A would work, but would it work as quickly or as readily? No. And substrate D, maybe if you're freaking starving for substrate, that would work, but then B never does, right? Does that make sense? So what I want to tell you about substrate specificity is that some substrates fit the active site better than other substrates. And so now with that in mind, I want to ask you, what is the relationship between substrate specificity and enzyme reaction rate? As substrate specificity increases, what happens to reaction rate? It increases, yes. So if you have exactly the perfect specific substrate, right, then your chemical reaction rate is going to be relatively high. Does that make sense to you guys? Okay, now, keep that one in your head because you have to contrast it with the next one that sounds very similar but has an important difference. And the important difference, oh, well, let me show it to you. So the next one is called enzyme substrate affinity. So specificity, right, is implying that there's a specific thing that I prefer above all others. What does affinity mean? What? Attraction, or how much you like it, right? I can say I have an affinity for champagne and red shoes and golden retrievers. It's all true, okay? So that is affinity, like how much you like something. So here's the deal with enzyme substrate affinity. You don't have the same enzymes that I do, okay? You probably have some of the same ones for the ones that like are super important, like cellular respiration enzymes, but you don't have all the same enzymes that I do, right? Your enzymes may have slight amino acid changes, a little bit of structural difference. So what happens is that if I have an enzyme that has low affinity for the substrate, then can you see it's going to take me a while for my reaction rate to go up, right? Because it doesn't bind the substrate very well. Does that make sense? It's going to take me longer to hit saturation. Overall, I'm going to generate less product over the same time period, right? But what if you have an enzyme that's just beautifully formed for the substrate and has a very high affinity for the substrate, right? Then it has a very fast chemical reaction rate. It binds to the substrate very effectively. It generates the product very effectively. It hits saturation pretty effectively. Now, the big difference is trying to put these two together. So substrate specificity, here's the difference, has to do with multiple possible substrates, yes? So within me, I have multiple possible substrates, depending on the amino acids, for instance, I've eaten. There's some amino acids that I might be using as part of a chemical process, and some of them are just slightly different than other ones, right? That makes sense? Multiple possible substrates. But enzyme substrate affinity has to do with multiple possible enzymes. Yes? Okay. And it's far more likely to vary between people, although it is distinctly possible that I have 2 copies of almost every enzyme, right? Because I have 2 parents, right? Okay, and one of them might work better. Now, if you are just a complete badass and your metabolic enzymes are working amazingly well, then you have a completely different metabolism than I do, right? Does that make sense? You can do things very, very effectively. Your enzymes are working really well. The interesting thing is effective from an evolutionary and metabolic standpoint is exactly the opposite of what we want. Historically speaking, hundreds of thousands of years of human evolution, would it really be an ideal thing for you to burn a cheeseburger as quickly as possible? No. The ideal thing is to have a nice, slow metabolism so that every bit of food is going to last you forever. So what you are battling against when you're trying to lose that last 10 pounds is 100,000 years of evolution. Maybe you should just buy some bigger pants. I don't know. So yeah, the metabolism, right, that you are going for, you're kind of, when you're trying to lose weight, going for a very inefficient metabolism, and your body is like, no, 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 that equals death. Something is going to eat you if you have a very inefficient metabolism, or you're just going to not survive the winter. So, do you see the difference between substrate specificity and enzyme substrate affinity? Okay, I said it. I want somebody to raise their hand and tell me. I'll be nice to you, I promise. Yeah, I know. You guys are going to have to like caffeinate or something in the afternoons because I'm done at 2:32, so one of us needs to be smart. So, okay, go ahead. You can do it. Go ahead.
So substrate affinity is having a liking to another substrate.
That's specificity. Substrate specificity is.
So the substrate specificity is basically stating that really get it perfectly. But then I have another that looks similar to it,
Mm-hmm.
and can go in, but may not work as well.
Right, that's substrate. That's enzyme substrate specificity. That's substrate specificity. And what about affinity? Can somebody tell me affinity?
Multiple enzymes.
Multiple different enzymes, same substrate. So, like every scientific experiment, you hold everything constant except for one variable, right? With one of them, what I'm saying is that you hold the enzyme constant and change the substrate. That's the first one, right? And the other one, you hold the substrate constant and change the enzyme, right?
So what was the substrate concentration then? That's how.
Substrate concentration is just how much substrate you have. That one corresponds to this. So as I increase substrate concentration, I increase the chemical reaction rate. Okay?
But on
Okay.
the affinity, then there are different enzymes?
With the, yes, they're different enzymes. You would have two different graphs, right?
Right.
That makes
So
sense?
that they could
For
just
two
grab
different
more
enzymes.
stuff.
So now let's go back to this and let's just do prediction about the chemical reaction rate for each of these situations. Can we do that? Right? So as you increase saturation level, you do what to the reaction rate? Increase the reaction rate, right? That makes sense? And that corresponds, for those of you who are visual people, that corresponds to this figure. Yes? Okay. Everybody good with that one? So that's saturation. Yes? Got it? Okay. Next one I want to do is substrate concentration. So you guys tell me, and you should know, as you increase substrate concentration, what happens to the chemical reaction rate? Chemical reaction rate goes up as you increase substrate concentration until you hit saturation. Good. All right, you're getting it. And then the next one is, we already did enzyme concentration. Did we do? No, we didn't do enzyme concentration. Okay, so the next one is as you increase the concentration of enzyme, what happens? Chemical reaction rate goes up. Okay, as you increase substrate specificity, what do you predict would happen, even though we don't have a graph for it? As you get, the chemical reaction rate would go up. As you increase enzyme substrate affinity, what would happen to the chemical reaction rate? Got it? Chemical reaction rate would go up. Okay, now this one is going to throw you for a loop, but we'll be all right with it.
Would they ever go down?
Right here.
Right now.
Okay, so right now, and I mean, if you did the opposite to any one of them, they would go down, right? I was just trying to be consistent as I was telling the story. Okay, so now what we're going to talk about is product concentration. And when we get to product concentration, I have to back up and tell a little bit of a story before we talk about it. So one of the things that we're going to see this time and next time is that generally when you're dealing with chemical reactions inside cells, you don't get a single chemical reaction for the product. A lot of times you need a series of chemical reactions until you get the final product. Does that make sense to you? And you generally, I mean, you know these, right? Cellular respiration and protein synthesis and all of that. It's not one chemical reaction, it's a whole bunch of them until you get the product. These are generally referred to as biochemical pathways, okay? So I'm just going to give you a fake one so that we can see what's going to happen. So we're going to start out with A plus B, and we're going to try to make C plus D. And then C plus D is going to go to E, and then E is going to fall apart to F plus G, and then ultimately what I'm trying to make is H, right? And each of these chemical reactions is catalyzed by an enzyme. Enzyme 1, enzyme 2, enzyme 3. You with me? Okay, so if I tell you that this is based on the principles of negative feedback, then you can probably predict what H should do to the rate of chemical reaction. You can probably predict, but let me show you sort of how it works. So we're going to sort of go with that same picture that we drew before, and we're going to draw enzyme 1, and enzyme 1 is going to have the same shape that we saw before. A is that shape, B is that shape. There's the active site, okay? That's where they're going to bind. This is enzyme 1, okay? But what happens is, via the principles of negative feedback, when I start getting enough H, it should cut off these chemical reactions, right? That makes sense? So what's going to happen is H is actually going to feed back via negative feedback inhibition, and it's going to bind, say H is round, okay? H is going to bind not to the active site, but to this site. Any site other than the active site could be considered a regulatory site. And then I'll give you two possibilities. When H binds to the regulatory site, it makes the active site fit the substrates better, or when H binds to the regulatory site, it makes the active sites fit the substrates worse. Exactly. Does that make sense? Do you see how if this worked over time, what you would see for the concentration of H over time is that when H was low, these chemical reactions were very active, right? And then H came up, right? And then as soon as H started increasing in frequency and concentration, it was going to be more likely to find the regulatory sites. And then H is going to go back down, and then it's going to go back up, and it's going to go back down. Okay, so some logic questions for you. Why is it less likely that H will feed back to enzyme 3? Why is that less likely than feeding back to enzyme 1?
Because you're trying to regulate.
What?
Because you're trying to regulate.
You are trying to regulate, but I want you to think about, I mean, both of them are possible, right? And both of them would accomplish ultimately the same thing.
One has more concentration.
It's not concentration.
Wouldn't
more
A,
fun.
B, and C, and D all keep happening?
Yeah, A and B and C and D would all keep happening. So you're going to end up wasting all of this potential energy, but at least components, right? You're going to either waste energy on the process, or at least you are going to make a bunch of unnecessary intermediate products, right? Does that make sense? So it's far more likely that the regulatory molecule, the product, will feed back toward the beginning of the biochemical pathway. Now, I'm not saying it might not feed back at multiple places, but it generally doesn't feed back just one step, because then you're going to keep doing all of the initial ones, right? So do you see that this is just a very small scale chemical example of a negative feedback inhibition loop, right? You get more product, it cuts off the reaction. As soon as you start running out of product, then remember, it's just statistics and likelihood, then there's going to be less product to actually find the regulatory sites, right? And then you get more enzyme on, and you get more product. And then you turn off the regulatory sites, right? And it just goes on and on and on. So you're regulating the level of product that you're generating. Does that make sense? So it's putting the chemistry together with the physiology. And a lot of times chemistry and physiology are a little conceptually difficult to put together, but this is one of the mechanisms by which you put them together. Okay, so product concentration. Often product concentration can regulate enzyme activity. It is negative feedback. So last question on this. What do you predict as product concentration goes up? What generally happens to the rate of chemical reaction? Generally goes down because of its negative feedback inhibition. That make sense? Okay, all right. Okay, we're going to stop there. So because the last class is a little behind you, what I'm going to do is the first quiz. It's going to be short, and it's going to be only over the first set of notes. They're behind you on this stuff. And then we'll just have to have another one once they catch up. Okay?
So the
That'll be on
chapter
Monday.
2 and 3 that we just started won't be on.
No, it won't be on there on Monday's quiz. But you do have a quiz on Monday.
Have a good one.
You too. Oh yeah, did everybody get right? You have to write your name on the clipboard with your clicker number before you leave. I have to register them before Monday. I will post this on the website, and there may be a note on there about what I want you to get ready before Monday too. Since the last class missed like 30 minutes of class, I may also put a worksheet up there that helps to like solidify the stuff that we just did, or get ahead of it. I can't figure out what I need to do yet.